Right. So Unit 1 is about the methods of biological science. And by that, we essentially mean we're talking about the scientific method. And as you completed the lab, the exercises that you did went over these different parts of the scientific method to get you more familiar with what this all entails and what it looks like when you are going about learning in the method that is scientific. And so there's pieces to this where when you are learning in this scientific method, the very first step is that you are going to be making observations. See, you are learning about your physical environment, about your surroundings. So in order to learn about your physical surroundings and your physical environment, you have to make observations. Now, this is not just visual observations, it's using all your senses. So things you hear, things you see, things you smell, all of this sensory input gets included in these observations that you make. And some key terms here, you need to understand the difference between inferences and assumptions. As you make your observations, you will be able to make some educated guesses about what you have observed. Those educated guesses are where you are making some sort of statement based on your observations. For example, in the lab, you should have looked at some skulls. And from these skulls, you would have made observations about their teeth, where those teeth are positioned in their jaws, the shape of the teeth, the way those teeth might move together, how they might, from the upper jaw and the lower jaw, they can possibly grind or rip or tear. And so these inferences are going to be evidence-based conclusions, that evidence being your observations about those skulls, so you can infer what kind of food an animal that had that skull and those teeth might eat based on the kind of movements that it can perform with those teeth, the shape of the teeth, whether it's good for ripping or tearing or whether it's good for grinding, those kinds of things. Assumptions are when you draw a conclusion like you do with an inference, but that conclusion is not based on your observations. You don't have evidence to back up your conclusion. So with the skulls example, that might be something like saying that the skull came from Columbia, Missouri. There is nothing in the skull itself that you can observe that would help you to draw that conclusion of where it came from. So that is an assumption. So your very first step of the scientific method is going to be making these observations and drawing inferences from those observations. From there, you can get to the questions. And the questions are now going to be things you want to kind of find out more about what you have observed and from these inferences that you're drawing from these observations. And in your questions, you're going to have both hypotheses and predictions. Hypotheses are what we call a causal statement. It is a statement where you are saying this is the cause for what I am observing or what I am predicting. Your prediction is then something that you think will happen if your hypothesis is correct. And we looked at hypotheses and predictions in the second exercise of the lab in the experiments that Reddy conducted, where he was testing this theory of spontaneous generation. And so in spontaneous generation, the idea is that living things appear spontaneously. And those living things that appear spontaneously do so when you follow a particular recipe to result in that particular life form appearing. So Reddy tested a specific recipe because it was one that he could test at home fairly simply. It was a recipe for flies, and it was simply having snake meat left out in the air, and you leave it for a particular amount of time and the flies appear. So the idea was that they appeared from the meat. Now, part of why people believed in spontaneous generation in Reddy's time is because they didn't have a lot of understanding of life cycles of insects. But they made these recipes for more than just insects. They made it for other animals too. And things that they didn't make a lot of observations about where they came from, how they arrived at different locations. They just would notice that they would appear in particular settings where it seemed like these recipes just made it so that they would show up. So he wanted to test that because he thought that this was wrong. He thought that the life had to come from some other source. It couldn't just appear on its own. And so you should have seen in the exercises how he tested this hypothesis. But the very first step was for him to form that hypothesis. He needed a causal statement. He needed something that he believed was the source of the life. And his hypothesis was that the life came from another source. They came from somewhere else and showed up in the meat. As opposed to the spontaneous generation hypothesis, which was that the life just appeared. And so the predictions are then where you are saying, if my hypothesis is right, this is what I expect will happen. So since Reddy was saying it was not spontaneous generation, that they had to come from somewhere else, then he was able to predict what would happen in the experiment that he set up, where he had some snake meat left on the counter and he had some snake meat in a sealed jar. Now, for those who believed in spontaneous generation, since the snake meat was the recipe, it shouldn't matter if it's on the table or in the jar. In either one, flies should appear. But since that would be their prediction, if their hypothesis was that spontaneous generation is where life comes from, that's the cause, they just appear, then in either of those situations, whether the snake meat is on the counter or in the jar, the flies will appear. But since Reddy said the flies have to come from somewhere else to show up in the meat, then the exposed meat that's just out on the counter, he says, we'll get the flies, because since it's just exposed, the flies can come from somewhere and end up on the meat, versus the one in the jar. The one in the jar is sealed, so it's preventing anything coming from somewhere else to end up on the meat. So that one has no flies, right? So this second exercise gives you this background with Reddy and with his experiment and lets you get to see how these hypotheses and predictions work. And an important part here when you're telling apart hypotheses and predictions is to make sure you look for that causal statement. Sometimes a lot of people learn that an if-then statement is a hypothesis, but you can write an if-then statement and have it just be a prediction and have it not be a hypothesis, because in order for it to be a hypothesis, it has to include that causal statement. It has to have something in there that says this is the cause of my observations. So something like if I have snake meat in a jar, no flies will show up because the flies have to come from somewhere else. Versus if it were just a prediction, then it would not have that causal statement. It would just say, when I put snake meat in a jar, no flies will show up. That's your if-then statement without the cause included. It has to include that cause in order for it to be a hypothesis. So then you get to test your predictions. So that's where the experimental design comes in. And we looked at that again more with Reddy. You got to see his experimental design for how he tested his hypothesis about spontaneous generation being wrong. And based on his experimental design, he made those predictions about where the flies would show up. Now, in experimental design, in order for it to work well, there's some important pieces here. An experiment that is designed well needs to have something that you can compare. That's where the control and the treatment come in. Your control group is one where you don't mess with it. You leave it be. You don't make any changes to it. And the treatment is the one where you're manipulating some variable. You're messing around with that and trying to see if the variable that you're manipulating will affect the outcome of the experiment. So for, again, falling back on the Reddy example, if he had his two groups. He had the snake meat that was open to the just sitting on the counter, and he had his snake meat in the closed jars. And so one of those is his control, and one is the treatment. The control is the one that he left alone, the one that he essentially followed that recipe for spontaneous generation without manipulating anything. So that'd be the one that was just left on the open counter. The treatment is then the one in the jar, because that's where he's now manipulating a variable. That variable he's manipulating is the access to the meat, right? The meat is now sealed off. It's not left in the open. He also had replication in his experiment. It shows you in the pictures that went along with the exercise that there were 4 jars for the sealed jars, and there were 4 bits of snake meat left out in the open on the counter. So this is your samples within your treatment. So the treatment group or the control group has to have multiple samples, because if something were to go wrong and you only had 1 sample, then you can't draw any conclusions from that. Just say, for example, the jar he used had holes in it that he didn't notice and flies showed up in there. Then he couldn't use that to draw any conclusions from his experiment. Or if the snake meat on the counter happened to have some kind of chemical in it that dissuaded the flies and they weren't attracted to it. And so no flies showed up even though it was left out on the counter. If he only had one piece of snake meat that he did that with, then again, he's can't draw any conclusions from this experiment. So you need that replication to help give your experiment more power, more weight behind it, that you can say, yes, this is definitely real because I did it multiple times. I had multiple pieces of meat and it still worked. And it worked for either all of my samples within my treatment or many of them. Okay. So I mentioned that you need that replication in case something goes wrong. So when things go wrong with an experiment, those are sources of error. And there's two kinds of error you need to be familiar with. You need to be familiar with sampling error and experimental error. Sampling error is when your sample is not representative of the actual population that you're looking at or the actual variable that you're looking at. And so sampling error is not a mistake. It's not something wrong with the experiment. It's just that the sample itself was not representative of the population. This gets looked at with the next exercise you guys did in the lab, where you looked at the shells and you had a random sample of shells, and that random sample of shells had different lengths that got measured of those shells. And the sample you got was 15 shells, and that was out of a population of, I think it's something like 200 some shells altogether that all these numbers are getting pulled from for your sample. So if you had gone and collected all the shells and taken the lengths of all the shells and averaged those, you would have come up with an average shell length that was the actual average of that population. You can't really do that very well in real life scenarios. In real life scenarios, you are trying to study a population, sometimes populations of birds or frogs or deer or something out there in nature that you want to know more about, and you can't go out there and catch every single individual of that population. It's just not going to happen. It's not possible to actually catch every individual in that population and get the data you want out of them. So you have to take a sample. And when you take a sample, if there's any kind of bias in the way you go about taking that sample, it might not be representative of the population. The way this could have happened with the shells is now for you guys, this was simulated on the computer and it gave you a random sample. But if you were physically out there collecting these shells and picking them up to go and take your lengths of these shells, if you felt like the big ones were too difficult to grab or too heavy or too hard to carry, then you only collected the small ones because they fit in your hands better. Well, that ended up biasing your sample. Your sample is not actually representative of the population. This can happen sometimes if what you're trying to catch is something you catch with a net, right? So if you cast your net to catch whatever it is you're collecting, but the size of the space in the netting is big enough that some things slip through, well, then you're only going to get the bigger ones and you're not going to get any of the smaller ones because the smaller ones slip through the net. and that's going to bias your sample. So things like that are what we talk about when we say sampling error. Experimental error is the one you're going to more typically think of when you think of errors happening in experiment. That's going to be things where you didn't measure correctly, you had, say, a scale that wasn't calibrated correctly, so all your measurements were off, or somebody made a mistake and measured in inches instead of millimeters, or somebody was off on their rounding and uh, they weren't rounding correctly when they took their measurements. Just anything like that that causes the experiment to not be accurate because you just did something wrong. That is experimental error. Okay, so far we've made some observations, asked some questions, and formed hypotheses and made predictions with those questions, and then come up with an experiment to test those predictions. We took some great care in our experimental design, making sure we had comparable groups with a control and a treatment, and that we replicated things to try to account for these possible sources of error. Then your experiment is done. You've collected all this data. You need to be able to understand your data. There's two measurements that tend to be very commonly used. These measurements are means and medians, and they're applicable for different situations. In situations where you have what's called a normal distribution, that's one that's the bell curve you might be familiar with, gets talked about with grades a lot, then it makes sense to use the mean. So if you got a normal distribution with that bell curve, it makes sense to use a mean, because the mean is your average, and in a bell curve situation, the average is kind of in the middle. That's where most of your individuals in the population are. You want to use medians when it's not a normal distribution, when your distribution is skewed one way or another. And that's because when you have a skew, you have somebody in that population is way, way different from everybody else. They're either way smaller or way bigger. And because of that, when you take your average, it throws your average off from what the actual typical individual in that population looks like. This is especially applicable for things like income, because when your income is very heavily skewed because there are a few people in the world, or in the United States, if you want to be specific to the US, that make lots and lots and lots and lots of money, like insane amounts of money. And because they make so much money, they skew the data and they throw everything off. And so if you were to calculate the mean for income, you would get a value that's much higher than what a typical person actually makes, because that 2 or 3 people that make lots and lots of money threw everything off. And so the median then is a more accurate value, because the median is the middle number. If you line them all up, smallest to largest, and you just start at either end, eliminating the smallest and the biggest, you work your way to the middle. That's your median. And that value is more representative of your population when you have skewed data. Okay, so these calculations tell you things about your population, but you have to make sure you know what your distribution in your population looks like to know whether you want to use your mean or your median. And then you're going to use graphs. Graphs help you visualize your data. It's not any good to just have a table full of numbers, because you can't really tell what's going on when you're just looking at those numbers. So you have to graph it, and you want to make sure you use the appropriate kind of graph, which is the next point. But first, I have here remembering to avoid extrapolation. So extrapolation is when you draw conclusions that don't fit within your data. So when you have data for, say, for example, your shells going from shells of size, say, 10 millimeters to 45 millimeters, just for an example. Those are going to be some small shells. If you don't have any shells that you measured that were 65 millimeters, that's outside that range of 10 to 45, then you draw some conclusions about something of that length. That's extrapolation because it's not within the range of your data. Interpolation is the word that goes with that. That's where you draw conclusions about something that is within your data. That's where you find a new shell that's a size within what you've already measured. Okay. So to know which kind of graph to use, you have to look at what kind of data you have, because you want different kinds of graphs for different kinds of data. Bar graphs are going to be used when you have categorical data. That's where your data has values that are words. So things like something sorted by color, or something sorted by species, or by type, something where you have to have some kind of descriptive word to say which category it falls in. So that categorical data is going to have bar graphs. Histograms are used for what we call frequency data. Frequency is when you're taking counts of something. So like you're saying, this is how many I have of this thing. And those counts can get sorted into ranges of groups. So say you're counting how many dogs fall within this weight range. So your frequency is the count, that's how many dogs, and then the range is the weights that you took of the dogs. So you can have dogs that weigh from 5 to 20 pounds, and then dogs that weigh from 20 to 30 pounds, and so on. And so you have these different ranges, and as long as the dog's weight is within that range, it goes in that spot on the histogram. And then line graphs are the ones you'll be the most familiar with in this course, because those are the ones you tend to have exercises where you graph data with line graphs. This is for continuous numerical data. Continuous numerical data is when you have numbers, that's numerical, that are not frequencies, right? Frequencies are counts, they're whole numbers. You can't have half of a dog, right? But a continuous number is one that can be a fraction, can be a decimal. So things like how much something weighs, how tall something is, how long something is, how fast something is, speed, acceleration, temperature, all of these values are continuous values. And you're going to have two of those values that you're comparing. It's some kind of relationship. like a relationship between height and weight, or a relationship between temperature and time. So these values, you are comparing them and you're saying that there is some sort of relationship between them where one value is dependent on the other. So those are a couple other terms from here. You have independent and dependent variables when you talk about line graphs. Those are one variable is the one you've manipulated. That's the independent one. And then the other is something that you are measuring that you are saying is affected by your manipulation. That's that relationship. And so that one is dependent on what you did to the independent variable. Okay. So for the graphs, this is the last bit I'm going to talk about here. I have a sample that helps explain these independent and dependent variables, where they go on the graph, and what it should look like when you do a line graph. For all of the graphs that you do in this course, you want to follow these rules. So anytime you graph something, it needs to have a title. It needs to have the axes labeled. That means you need to describe what is on the axis. And that description should include the units. So like here we have, in this example, your heating time in minutes. So what we have in parentheses here, the M stands for minutes. That's our unit. So we're saying this is how many minutes we spent heating this object. And then over here we have temperature, and it's in degrees Celsius. That's our unit. So our labels include our units. It needs to be the proper form. That means that you've chosen the right kind of graph to do. So if it should be a histogram, it needs to be a histogram. If it should be a bar graph, it needs to be a bar graph. So that's where it's important to check what kind of data you have and make sure you're doing the correct type of graph. And then if it is a line graph, you need to be doing a line of best fit. A line of best fit is one that follows the trend in the data. Here, it's just a nice straight line straight through these data points. But sometimes your data is kind of scattered, or it goes kind of up and down a little bit, right? So like this might be higher and this might be lower, and so on. And you end up with something that if you just drew straight through the dots, you would have a zigzaggy line. You don't want to do that. That's not a line of best fit. That is connecting the dots, connecting your data points. But what you want is to have this line showing what trend is the data following, which direction does it seem generally to be going. And so if it's kind of zigzaggy on the dots, rather than following those dots and drawing a line through each one, you would draw still this straight line that goes through, showing the direction the data tends to be going or is trending towards. Additionally, you don't want to draw past the data you've actually collected. That's that extrapolation that we talked about. So in this example, this person collected data from time point 0 to 50. If they extended the line past this dot, that would be extrapolation. And if they extend it past this dot, that would be extrapolation. You don't want to do that. Additionally, this is a good place to see the difference between extrapolation and interpolation visually. So this person finished their experiment in 50 minutes. if they wanted to try to make some kind of prediction of what temperature would this object that I heated be if I had left it on there for 70 minutes, they would have to extend the line past the data they collected to make that prediction. They can't do that. That's extrapolation. That's what we're avoiding and we don't want to do. And you avoid that because trends don't always hold up past whatever point you collected in your data. If this was, for example, heights and weights instead of time and temperature, you don't usually see a straight line continue on indefinitely for that, right? As somebody gets bigger, they don't keep getting taller. You can only get so tall before you can't grow anymore. So it doesn't work. Additionally, it doesn't work to have negative time, right? You can't go backwards in time, so it doesn't make sense to go past the zero in this direction. So you can't go past your data points. But interpolation would be something like saying, well, I didn't collect data at 25 minutes, but I have a pretty good guess that when my object was being heated for 25 minutes, that it was about 45 degrees Celsius, because my line goes through that point. Okay, so that's interpolation. That's where, even though I didn't collect data at 25 minutes, I can say pretty comfortably that I think it was about 45 degrees when it was being heated for 25 minutes. So that's the interpolation. So that's everything I wanted to go over on Unit 1. And there are some additional slides for you to look at that go over the learning objectives in more detail down below.